On January 23, 1960, two men climbed inside a small steel sphere and sank into the Pacific Ocean. They were Jacques Picard, a Swiss oceanographer, and Don Walsh, a lieutenant in the United States Navy, and they were aboard the Bathyscaphe Trieste, a vessel designed to go deeper than any human being had ever gone. They descended for 4 hours and 48 minutes, watching the pressure gauges climb and the temperature drop and the light fade from blue to gray to absolute black. And then, at a depth of 10,916 meters, the steel sphere touched the sediment of the Challenger Deep, the lowest point in the Mariana Trench, the deepest place on the surface of the Earth. They spent 20 minutes on the bottom. Through the porthole, they could see almost nothing. A pale sediment stirred by their arrival, a few shrimp visible in the beam of their lights. Picard later reported seeing what appeared to be a flatfish resting on the sediment, an observation that would be debated for decades, because a flatfish at 11 kilometers depth seemed to violate everything biologists believed about the limits of vertebrate life. Then they began their ascent, returning to the surface 9 hours and 15 minutes after leaving it, having spent less than half an hour in the deepest place on Earth. Subscribe now and come back every week. Every documentary is designed for the hours before sleep, calm and unhurried, always going somewhere you have never been. 65 years later, the Challenger Deep has been visited by fewer people than have walked on the surface of the Moon. The technology required to reach it is more demanding than the technology required to launch a satellite. The pressures at that depth, approximately 11,000 atmospheres, would crush any conventional submarine in seconds. The cold is not the cold of winter, but the cold of water near its freezing point, compressed to densities that make it behave almost like a different substance. And yet the Mariana Trench is not empty. It is alive, in ways that biologists are still struggling to understand, with organisms adapted to conditions that should, by any reasonable extrapolation from surface biology, be incompatible with life. The Mariana Trench is located in the western Pacific Ocean, to the east of the Mariana Islands, which are themselves the peaks of a volcanic arc produced by the subduction of the Pacific Plate beneath the Mariana Plate. The trench is approximately 2,550 kilometers long and 69 kilometers wide at its broadest point, though its width varies considerably along its length. It is crescent-shaped, curving southward from the northern Mariana Islands toward Guam. And its deepest point, the Challenger Deep, is located in the southern portion of the trench, near the island of Guam, at coordinates that have been measured with increasing precision by successive surveys and that are now known to within a few meters. The depth of the Challenger Deep, which has been measured as ranging from 10,924 meters to 11,034 meters depending on the method and the location of the measurement, makes it the single most extreme topographic feature on Earth. If you placed Mount Everest at the bottom of the Challenger Deep, its summit would still be more than 2 kilometers below the surface of the ocean. The volume of water above the deepest point of the Mariana Trench is literally incomprehensible. The entire atmosphere, with all its weather and its gases and its blue expanse visible from space, is less massive than the column of water pressing down on the sediment at that depth. The weight of the water column above the Challenger Deep is almost impossible to convey in terms that feel real. The total mass of water pressing down on every square centimeter of the deepest seafloor is equivalent to the weight of approximately 50 elephants stacked on a surface the size of a postage stamp. Materials that are rigid and unyielding at surface pressures, including many metals and ceramics, become plastic and deformable at those pressures. Gas-filled spaces collapse instantaneously. Biological membranes that are carefully maintained at precise tensions in surface organisms are compressed into configurations that prevent them from functioning as the selective barriers that make cellular life possible. The engineering challenge of building a vessel that can maintain 1 atmosphere of internal pressure while the external pressure is more than 1,000 times higher is one of the most demanding in the history of materials science, and the solutions that have been developed for deep-sea submersibles represent some of the most sophisticated pressure engineering in existence. The geological origin of the Mariana Trench is the same process that produces all the ocean trenches on Earth. Subduction, the descent of one tectonic plate beneath another into the hot mantle below. The Pacific Plate, which is the largest tectonic plate on Earth and which has been moving westward and northward across the Pacific Basin for hundreds of millions of years, encounters the Mariana Plate along the line of the Mariana Trench. The Pacific Plate is older and denser than the Mariana Plate, and so it descends beneath it rather than riding over it. As the Pacific Plate bends downward and begins its journey into the mantle, it creates the deep depression that is the trench, a place where the seafloor has been pulled downward by the weight of the descending slab, and where the bending of the oceanic crust creates a topographic trough of extraordinary depth. The Pacific Plate at the Mariana Trench is approximately 170 million years old, making it some of the oldest oceanic crust on Earth. Oceanic crust ages as it moves away from the spreading center where it was created, cooling and contracting and becoming progressively denser. The older the oceanic crust, the denser it is relative to the mantle beneath it, and the more readily it descends into the mantle when it reaches a subduction zone. The extreme age of the Pacific Plate at the Mariana Trench is one of the reasons the trench is so deep. Old, dense, cold oceanic crust descends steeply, pulling the seafloor down to greater depths than young, buoyant crust would. The angle of subduction at the Mariana Trench is nearly vertical in some places, one of the steepest subduction angles known anywhere in the world. That steep angle reflects the high density of the subducting slab and produces a trench that is not only deep but narrow, with steeply inclined walls on both sides. The seismic surveys that have mapped the structure of the trench in detail show the subducting Pacific Plate disappearing into the mantle at an angle of close to 90 degrees in the deepest portion, essentially dropping straight down rather than sliding gradually. The scale of the Mariana Trench relative to the rest of the ocean system is worth contemplating before considering the life within it. The average depth of the world's oceans is approximately 3,800 meters. The Challenger Deep is nearly 3 times that depth. If you were to drain all the water from the Earth's oceans, the resulting landscape would include the vast flat abyssal plains at 4 to 5 kilometers depth, the mid-ocean ridges rising to 2 or 3 kilometers depth, the continental shelves at less than 200 meters, and then, in the western Pacific, a narrow crescent-shaped gash descending to 11 kilometers, deeper than everything else by a margin that exceeds the height of the Himalayas. The Mariana Trench is not just deep, it is anomalously, dramatically, almost impossibly deep even by the standards of the deep ocean. The volcanic arc that produces the Mariana Islands is the surface expression of the subduction process. As the Pacific Plate descends into the mantle, it carries with it water and other volatile compounds that were incorporated into the oceanic crust and the sediments above it. At depths of approximately 100 to 150 kilometers, the pressure and temperature are sufficient to drive those volatiles out of the slab and into the overlying mantle wedge, where they lower the melting point of the rock and produce magma. That magma rises through the overlying plate and erupts at the surface, building the chain of volcanic islands that parallels the trench at a distance of approximately 200 kilometers. The floor of the Mariana Trench is not flat. The deepest portions of the trench, including the Challenger Deep, are actually depressions within the larger trench floor, separated from each other by ridges and elevated areas that divide the trench into a series of basins. The Challenger Deep itself consists of 3 separate pools, known as the Eastern, Western, and Central Pools, each slightly different in depth and each separated by shallow sills that restrict the exchange of water between them. The deepest water in each pool is essentially trapped below the sill depth, isolated from the rest of the ocean for very long periods and developing its own distinctive chemical and biological character. The physical conditions at the bottom of the Mariana Trench are as extreme as any environment on Earth's surface, and understanding them is essential for understanding the life that has adapted to survive there. The discovery of the Challenger Deep itself is a story that reflects the technological limitations of 19th-century ocean science. HMS Challenger, the British naval corvette that undertook the first systematic oceanographic survey of the world's oceans between 1872 and 1876, made the first sounding of the deep trench that would eventually bear its name during its traverse of the western Pacific. The sounding was made using a hemp line weighted with iron, a technique that could determine the depth of the ocean but could tell nothing about what lay at the bottom. The depth recorded by the Challenger's crew, approximately 8,184 meters, was a significant underestimate of the true depth of the trench, partly because of the difficulty of measuring such extreme depths with available technology and partly because the Challenger did not happen to sound the very deepest point. But the recording of a depth greater than any previously measured in that region of the ocean established the existence of an exceptionally deep feature, and the trench and its deepest point were named in honor of the ship and its mission. The pressure at the Challenger Deep is approximately 1,100 atmospheres, or about 110 megapascals. To put that in perspective, the pressure at sea level is 1 atmosphere. The pressure in a deep-sea submarine is maintained at 1 atmosphere by the vessel's pressure hull. The pressure in the deepest part of the Mariana Trench is more than 1,000 times that. A human body at that pressure, without a pressure suit, would be crushed instantaneously. The bones would shatter. The gas-filled spaces in the body, including the lungs, the sinuses, and the digestive tract, would collapse. The body would be reduced to a mass of compressed tissue in milliseconds. That pressure affects water in ways that matter for the chemistry and biology of the trench. Water under high pressure has a slightly higher density than water at atmospheric pressure, which is why the water at the bottom of the Mariana Trench is technically denser than the water at the surface. It also has a slightly lower temperature of maximum density, which affects the way the water circulates. And the solubility of gases in water increases with pressure, which means that the deep water of the Mariana Trench is supersaturated with oxygen relative to what would be expected at the same temperature at lower pressures. That elevated oxygen solubility is part of why aerobic life can exist at those depths despite the absence of photosynthesis. The temperature at the bottom of the Mariana Trench is approximately 1 to 4 degrees Celsius, just above the freezing point of seawater. That cold reflects the origin of the water at those depths. It is primarily Antarctic bottom water, the cold dense water produced around the Antarctic continent and spreading northward along the ocean floor. The cold, combined with the high pressure, creates a physicochemical environment unlike anything at the ocean surface. The enzymes and membranes of surface organisms would cease to function in those conditions. The organisms of the Hadal zone have evolved biochemical adaptations that allow their cellular machinery to continue working despite the extreme cold and the crushing pressure. The darkness at the bottom of the Mariana Trench is absolute. The last photons of sunlight are absorbed within the upper few hundred meters of the ocean. At 11 kilometers, there has been no sunlight for as long as the water has been there. The only light at the bottom of the Mariana Trench is bioluminescence, the light produced by living organisms through chemical reactions in their bodies. That biological light, which in the deep ocean is used for communication, predation, and defense, is the only illumination in an environment that has been dark since before the Atlantic Ocean existed. The chemical environment of the trench floor is also unusual. The sediment that accumulates in the deepest portions of the trench is a mixture of material that has sunk from the surface ocean above, including the skeletal remains of planktonic organisms, organic particles from dead organisms, and mineral particles from distant continental sources. That organic material, concentrated by the funnel shape of the trench which focuses settling particles from a wide area into a small depression, provides a food source for the organisms living there. But it also creates a reducing chemical environment in the sediment, where oxygen is consumed by bacterial decomposition and sulfate reduction produces hydrogen sulfide, creating chemical gradients that support chemosynthetic communities. The life that exists in the Mariana Trench is one of the most extraordinary discoveries of modern marine biology, and the exploration of that life is still in its earliest stages. The Hadal zone, defined as the water column and seafloor below 6,000 meters, was long assumed to be essentially lifeless or nearly so. The combination of extreme pressure, cold, and darkness seemed to preclude the existence of complex organisms. The first systematic biological sampling of the Hadal Zone, conducted by the Galathea Deep Sea Expedition in 1951, produced a dramatic surprise. Organisms were found at depths previously thought to be uninhabitable, including sea anemones, polychaete worms, sea cucumbers, and isopods. The discovery forced a complete revision of the models of deep-sea biology and opened a new field of research that continues to produce unexpected findings. The thermodynamics of the Mariana Trench environment place stringent constraints on what kinds of metabolic strategies can support life there. All life requires energy, and in the absence of sunlight, the only sources of energy available in the Hadal Zone are the organic material sinking from the surface and the chemical energy available from the oxidation of reduced compounds such as methane, hydrogen, and sulfide. The efficiency with which organisms can extract energy from those sources is limited by the thermodynamics of the chemical reactions involved, and the cold temperature of the Hadal Zone slows all chemical reactions, including metabolic ones, by factors of 2 to 3 for every 10 degrees of cooling. Life in the Hadal Zone therefore operates at a greatly reduced metabolic rate compared with life in warmer environments, a fact that is reflected in the slow growth, low reproductive rates, and extreme longevity of many Hadal organisms. The first organisms to be collected from the Hadal Zone were retrieved by accident, caught in the nets and trawls of ships that were sampling at depths they considered to be the limits of the ocean floor. The realization that those organisms had come from even greater depths than intended, that the nets had somehow descended beyond their intended operational depth, produced initial disbelief among the biologists who examined the specimens. The concept that life could exist at depths of more than 6,000 meters was simply not part of the scientific worldview of the time. The gradual accumulation of specimens from ever greater depths, through increasingly systematic sampling expeditions, eventually forced the recognition that the Hadal Zone was not lifeless but was instead a poorly known environment inhabited by a specialized fauna that had been present there for far longer than biology had been paying attention. The organisms most commonly associated with the Hadal Zone are amphipods, small crustaceans distantly related to shrimp that have proven to be astonishingly abundant and diverse in the trenches of the world's oceans. The Mariana Trench amphipods can be found in densities of thousands of individuals per square meter in some locations, feeding on the organic material that sinks from above and in turn being consumed by larger predators. Some species of Hadal amphipods can reach lengths of 30 centimeters, significantly larger than their relatives in shallower water, a phenomenon known as deep-sea gigantism that may be related to the cold temperatures and abundant food supply in some trench environments. The sea cucumbers of the Mariana Trench are perhaps the most visually striking Hadal organisms. Echinoderms in the class Halethuaroidea, they are elongated, soft-bodied animals that crawl along the seafloor ingesting sediment and extracting the organic material from it. In the Challenger Deep, sea cucumbers of the genus Halethuria have been observed in such abundance that they appear to carpet the sediment in some areas, their pale translucent bodies moving slowly across the seafloor in what has been described by researchers as a kind of slow-motion ballet. Those sea cucumbers represent the dominant megafaunal organisms at the bottom of the deepest place on Earth. Polychaete worms, the segmented marine worms that are found in virtually every marine environment from intertidal zones to the deepest trenches, are also abundant in the Mariana Trench. Some Hadal polychaetes have been found to be among the largest of their kind, reaching lengths of several decimeters, while others are microscopic infaunal species living in the sediment. The diversity of polychaete species in the Mariana Trench is only beginning to be characterized, and each sampling expedition that retrieves sediment cores from the trench floor produces species new to science. Bacteria are present in the Mariana Trench in extraordinary abundance, and their ecological role in the Hadal Zone is fundamental. Bacterial decomposition of the organic material that reaches the trench floor processes most of the carbon input to the Hadal ecosystem, recycling nutrients and creating the chemical gradients that support other organisms. Piezophilic bacteria, those adapted to high-pressure conditions, have been isolated from Mariana Trench sediments and studied in pressure chambers that simulate the conditions of the trench floor. Those organisms have revealed molecular adaptations that allow their cellular machinery, including their membranes, enzymes, and ribosomes, to function at pressures that would inactivate the equivalent structures in surface bacteria. The question of what fish live in the Mariana Trench is one that has been answered in stages as technology has improved, and the answers have consistently surprised the scientists studying the trench. The snailfish of the family Leparidae have emerged as the dominant vertebrates of the Hadal zone, found at greater depths than any other fish. In 2014, a research team using deep-sea lander cameras in the Mariana Trench observed snailfish at depths of approximately 8,143 meters, the deepest fish observation ever recorded at that time. Subsequent expeditions pushed that record deeper, and in 2023, a research team reported observing a snailfish at a depth of approximately 8,336 meters in the Mariana Trench, which became the new record for the deepest fish ever seen. The Hadal snailfish are remarkable animals. They are pale and translucent, their internal organs visible through the skin, giving them an almost ghostly appearance in the white light of the cameras. They are surprisingly agile for creatures living at such depths, swimming actively through the water column around bait deployed by the lander cameras. And they are apparently successful predators. Their stomachs, when retrieved and analyzed, contain the remains of amphipods and other crustaceans, indicating that they are active hunters rather than scavengers. The evolutionary origins of the Hadal amphipods, which are the most abundant and most diverse macrofaunal organisms in the Mariana Trench, have been studied through molecular phylogenetic analysis of their DNA. Those studies show that the Hadal amphipods are not a single group of organisms that descended from a single colonization event, but rather a polyphyletic assemblage, meaning that different Hadal amphipod lineages have descended from different non-Hadal ancestors that independently colonized the Hadal zone at different times. That pattern of independent colonization, repeated multiple times across the evolutionary history of the group, suggests that the Hadal zone has been colonized from shallower environments repeatedly over geological time, with each colonization event followed by adaptation to the extreme conditions and eventual radiation into new species within the Hadal zone. The molecular clock analysis of those evolutionary events suggests that some Hadal lineages have been in the Hadal zone for millions of years, while others may be more recent arrivals. The adaptations of the Hadal snailfish to their extreme environment include biochemical modifications that are being studied intensively by researchers interested in how life can function at high pressure. The cell membranes of snailfish are enriched in unsaturated fatty acids that maintain their fluidity at high pressure and low temperature, preventing the rigidification that would occur in membranes with more saturated fatty acids. The proteins of the snailfish, including their enzymes and structural proteins, have amino acid compositions that maintain their flexibility and function under the compressive force of the deep. And their bones, which in surface fish are largely mineralized calcium phosphate, are reduced in the Hadal snailfish to a more cartilaginous structure that may be better able to withstand the pressure or may simply reflect the reduced energetic cost of building hard structures in an environment where calcium is limiting. The earlier observation by Jacques Picard of a flatfish at the bottom of the Challenger Deep in 1960 has been revisited in light of what is now known about Hadal fish. Most modern biologists believe that what Picard saw was probably a sea cucumber rather than a flatfish, a conclusion consistent with the abundance of those organisms at Hadal depths and the limited optical quality of the observations available to him through the bathyscaphe porthole. True flatfish, which belong to the order Pleuronectiformes, have not been reliably recorded at depths greater than about 8,000 meters, and the biological adaptations required to maintain a vertebrate body plan at the pressures of the Challenger deep floor would be extreme. But the question of the maximum depth at which vertebrate life can exist remains open, and each new deep-sea expedition tests the boundaries of what is known. The acoustic environment of the Mariana Trench has also been studied with increasing sophistication. Sound travels differently in the deep ocean than at the surface. The higher pressure and lower temperature affect the speed of sound in ways that create acoustic focusing layers that can trap sound and allow it to travel extraordinary distances. The hydrophone recordings made in the Mariana Trench have captured a complex soundscape that includes the sounds of ship traffic thousands of kilometers away, the vocalizations of whales, the rumble of earthquakes, and sounds of biological origin that are not fully understood. The trench is acoustically connected to the surface world in ways that the visual disconnection of the deep does not suggest. The exploration history of the Mariana Trench is a story of technological innovation driven by the desire to reach and understand one of the most inaccessible places on Earth. The nitrogen cycle in the Mariana Trench sediments is one of the most important and least understood aspects of the trench's biogeochemistry. Nitrogen is an essential element for all life, and its cycling between organic forms, inorganic dissolved forms, and gaseous forms shapes the productivity of marine ecosystems. In the Hadal sediments, the decomposition of organic nitrogen by bacteria releases ammonium, which can then be converted to nitrate by nitrifying bacteria or reduced to nitrogen gas by denitrifying bacteria. The relative rates of those processes in the extreme conditions of the Hadal zone are not well constrained, but they affect both the availability of nitrogen for biological production and the export of fixed nitrogen from the Hadal zone to the broader ocean system. Understanding the nitrogen cycle of the Hadal zone is therefore important both for the local ecology of the trench and for the global biogeochemical cycles that regulate ocean productivity. After the Trieste's historic dive in 1960, more than 50 years passed before the Challenger Deep was visited again by a crewed vessel. In March 2012, filmmaker and explorer James Cameron descended to the bottom of the Challenger Deep in a specially designed submersible called Deep Sea Challenger, spending approximately 3 hours on the seafloor. Cameron's dive used technologies that had not existed in 1960, including high-definition cameras, LED lighting systems, and a pressure sphere made of syntactic foam rather than the massive steel of the Trieste's float. He reported a barren, almost lifeless environment at the bottom, noting that he saw very few organisms during his time there. That observation, which seems inconsistent with the biological surveys conducted by remote landers, may reflect the disturbance caused by the submersible's thrusters or the limited time and field of view available. The limiting factor, a submersible designed and operated by Triton submarines, has made more dives to the bottom of the Challenger Deep than any other vehicle in history, as part of the 5 Deeps expedition in 2019 and subsequent commercial and scientific expeditions. The 5 Deeps expedition, led by Victor Vescovo, successfully reached the deepest point in each of the world's 5 oceans and produced more detailed measurements of the Challenger Deep's depth than had previously been available. The expedition also recovered biological and sediment samples that have been studied by marine biologists and geochemists. The uncrewed lander systems that have become the primary tools for biological exploration of the Mariana Trench represent a different approach to deep-sea exploration than crewed submersibles. A typical deep-sea lander is a steel frame carrying cameras, lights, bait containers, and sample collection devices, dropped from the surface ship and sinking to the bottom under its own weight. When the mission is complete, a release mechanism, activated either by a timer or by an acoustic command from the surface, drops the ballast weight and the lander floats back to the surface. Those landers can carry biological traps, sediment corers, and environmental sensors that provide far more detailed measurements of the trench environment than a single crewed dive could achieve. The chemical composition of the water in the Mariana Trench reveals the long-term geochemical processes that shape the Hadal environment. The concentration of dissolved silica in the deep water of the trench is significantly higher than in the surrounding ocean, reflecting the dissolution of the siliceous skeletons of diatoms and radiolarians that accumulate on the trench floor and dissolve under the high-pressure, low-temperature conditions. The concentration of dissolved barium, an element that is scavenged from seawater by biological processes and released during the remineralization of organic matter in the sediments, is also elevated in the deep water of the trench, reflecting the high rate of organic matter input and decomposition. Those chemical signatures of the trench water provide information about the intensity of biological processes within the Hadal zone that complements the direct biological observations. The data from those lander deployments have revealed that the biological activity at the bottom of the Mariana Trench is highly variable in space and time. Some deployments in the Challenger Deep have recorded abundant fauna, including dense aggregations of amphipods attracted to the bait within minutes of the lander reaching the bottom. Others, in slightly different locations or at different times, have found much lower abundances. That variability may reflect the patchy distribution of organic input from the surface, the different sediment types on the trench floor, or temporal variability in biological activity related to the seasonality of production in the surface waters thousands of meters above. The Japanese research vessel Kairei and its remotely operated vehicle Keiko made multiple dives to the Challenger Deep between 1995 and 2003, collecting the most comprehensive set of biological and sediment samples from the deepest part of the trench that had been obtained up to that point. The Keiko samples revealed a microbial community of extraordinary diversity in the sediments of the Challenger Deep, with bacterial diversity comparable to that found in productive shallow marine environments despite the extreme physical conditions. That diversity challenged the assumption that extreme environments necessarily support low-diversity communities, and it suggested that the Hadal zone, far from being a biological wasteland, is an environment that has been colonized by a wide range of organisms over the long history of the subduction zone that created it. The chemistry of the water in the Mariana Trench reveals a world that is connected to the rest of the ocean in complex ways, despite its extreme isolation. The deep water of the Mariana Trench is not stagnant. It circulates on timescales of decades to centuries, driven by the density differences between the cold, salty water that flows into the trench from the surrounding deep ocean and the water that is modified by biological and chemical processes within the trench itself. That circulation, which has been studied using measurements of dissolved oxygen, temperature, salinity, and the concentrations of various chemical tracers, shows that the deep water of the Challenger Deep was last in contact with the surface ocean approximately 300 to 500 years ago, meaning that the oxygen dissolved in it was incorporated into the water during the reign of European monarchs whose names are now taught in history classes. The concentration of dissolved oxygen in the Challenger Deep is surprisingly high, despite the age of the water and the metabolic activity of the organisms living there. That high oxygen concentration reflects the combined effects of the high solubility of oxygen at low temperatures and high pressures and the relatively slow rate of oxygen consumption by the sparse biological community. The abundance of oxygen is one of the factors that makes aerobic life possible at Hadal depths despite the absence of photosynthesis, and it explains why the Hadal zone, far from being anoxic, supports a diverse community of aerobically respiring organisms. The organic carbon that reaches the bottom of the Mariana Trench comes from multiple sources. Some of it is the remnant of material that sank slowly from the surface ocean over months or years, progressively degraded by bacterial decomposition as it descended but not completely consumed before reaching the bottom. Some of it is more recent material from events of episodic productivity at the surface, such as the settling of phytoplankton blooms that produced large quantities of organic material that sank rapidly before it could be fully degraded. And some of it is transported laterally from the slopes of the trench walls, where the movement of sediment by gravity and by the circulation of the deep water brings material from shallower environments into the deepest basins. The concentration of persistent organic pollutants in the Mariana Trench is one of the most disturbing findings of recent deep-sea research. Studies published in 2017 and 2018 reported elevated concentrations of polychlorinated biphenyls, or PCBs, and other chlorinated organic compounds in the tissue of amphipods collected from the Mariana Trench. Those compounds, which were widely used as industrial fluids and electrical insulators before their production was banned in most countries in the 1970s and 1980s, had apparently been transported to the bottom of the deepest ocean by the sinking of organic matter and by direct uptake through the food chain. The concentrations found in some Mariana Trench amphipods were higher than those found in amphipods from polluted coastal environments, demonstrating that human industrial contamination has reached even the most remote and inaccessible place on Earth. The distribution of life within the Mariana Trench is not uniform with depth. The transition from the abyssal zone, below 4,000 meters, to the hadal zone, below 6,000 meters, marks a significant change in community composition and abundance that reflects the increasing extremity of the conditions. At intermediate hadal depths, between 6,000 and 9,000 meters, the community includes a diverse assemblage of amphipods, polychaetes, sea cucumbers, and other metazoans that is broadly similar in composition to the communities of abyssal environments, though with some distinct hadal-adapted species. Below 9,000 meters, the community changes again. The diversity of metazoans decreases, the dominance of amphipods and sea cucumbers becomes more pronounced, and the xenophytophores and other very large single-celled organisms become more prominent. At the very bottom, in the deepest basins of the Challenger Deep, the community is reduced to the most pressure-tolerant of the hadal organisms. The interaction between the descending Pacific Plate and the overlying Mariana Plate at the subduction zone generates not only the trench itself but also a complex array of geological features in the forearc, the region of seafloor between the trench and the volcanic arc. The forearc of the Mariana system includes a series of elevated ridges and depressions, the most notable of which is the Mariana forearc serpentinite seamounts, large dome-shaped features that are composed primarily of serpentinite, a green rock formed by the hydration of ultramafic mantle rocks by the fluids expelled from the subducting slab. Those seamounts are of considerable scientific interest because they expose mantle material that is normally inaccessible and because the fluids they expel create unusual chemical environments that support distinctive biological communities. The conical seamount and South Chamorro seamount, both of which have been extensively studied by ocean drilling expeditions, have provided important insights into the fluid processes associated with subduction and into the biology of serpentinite-associated ecosystems. The microplastic contamination of the Mariana Trench has been documented by several research groups since the mid-2010s. Plastic fragments and fibers have been found in the water column of the trench at all depths sampled, in the sediments of the trench floor, and in the gut contents of amphipods and other organisms collected from the hadal zone. The trench acts as a sink for microplastics, which accumulate there because of the convergence of ocean currents that funnel floating material toward the subduction zone and because of the settling of denser plastic particles through the water column over time. The persistence of microplastics in the hadal environment and their incorporation into the food chains of organisms living at the deepest point on Earth is a sobering illustration of the global reach of plastic pollution. The biology of the Mariana Trench has connections to some of the most fundamental questions in the science of life, including the question of the origin of life itself. The hydrothermal systems associated with subduction zones are different from the mid-ocean ridge hydrothermal systems that have attracted most scientific attention since their discovery in the late 1970s. Mid-ocean ridge hydrothermal vents are high-temperature, low-pH systems driven by the interaction of seawater with hot newly erupted volcanic rock. The hydrothermal systems near subduction zones, including those in the Mariana region, are influenced by the fluids released from the subducting slab and tend to be lower in temperature, higher in pH, and enriched in methane and hydrogen rather than in the sulfide compounds that fuel the chemosynthetic communities of mid-ocean ridge vents. The serpentinization systems of the Mariana forearc, where seawater reacts with ultramafic rocks in the forearc region to produce hydrogen, methane, and high pH fluids, are of particular interest to researchers studying the origin of life. The chemical conditions of those systems, alkaline, hydrogen-rich, and moderate in temperature, have been proposed as analogous to the conditions under which the first life on Earth may have arisen, based on the alkaline hydrothermal vent hypothesis developed by the geochemist Michael Russell and others. Under that hypothesis, the energy gradient between alkaline hydrothermal fluids and the acidic, carbon dioxide-rich water of the ancient ocean drove the prebiotic chemistry that eventually produced the first self-replicating molecules and the first protocells. The Challenger Deep is also connected to the question of the limits of the biosphere, the total inhabited volume of the Earth. The discovery of living organisms at the bottom of the Mariana Trench extended the known lower boundary of the biosphere by more than a kilometer compared with earlier estimates, and subsequent research has found living organisms in the subsurface sediments of the trench at depths of several meters below the seafloor surface, adding further depth to the inhabited zone. The total extent of the deep subsurface biosphere, including the sediments of the ocean floor and the fractures in the oceanic and continental crust, is now estimated to contain approximately as much biomass as all the organisms living on the surface of the Earth combined. The resilience of life in the Mariana Trench is relevant to the search for life elsewhere in the solar system. The most promising candidates for extraterrestrial life in the solar system are the subsurface oceans of icy moons such as Europa and Enceladus, which are likely cold, dark, and under high pressure in ways that are broadly analogous to the conditions in the Hadal Zone. The organisms that have adapted to the Mariana Trench environment demonstrate that life can thrive in cold, dark, high-pressure environments as long as energy and chemical resources are available. That demonstration strengthens the case for life in the subsurface oceans of the outer solar system, and the biochemical adaptations of Hadal organisms provide a model for what that life might look like at the molecular level. The xenophyophores, giant single-celled organisms found at Hadal depths in the Mariana Trench, are perhaps the most extreme example of unicellular life on Earth. Xenophyophores are foraminiferans, relatives of the tiny shell-building organisms that dominate the deep-sea sediment record, but they grow to sizes of up to 20 centimeters, making them among the largest single cells known. They build elaborate tests or shells from sediment particles glued together with organic cement, creating complex three-dimensional structures on the trench floor. A single xenophyophore cell can contain millions of nuclei, a condition called multinucleation that allows the organism to grow to its extraordinary size while maintaining the biochemical efficiency of a single cell. They are strange and magnificent organisms, and their discovery in the Mariana Trench was one of the most unexpected findings of Hadal biology. The sound produced by the Mariana Trench itself is a subject that has attracted increasing scientific interest. The convergence of two tectonic plates at the Mariana subduction zone is not a silent process. The seismic activity associated with the subduction produces a continuous background of low-frequency vibrations that are detectable by hydrophones deployed in the trench. Those seismic signals include the low-frequency tremor associated with the gradual deformation of the descending slab, the higher frequency signals from small earthquakes on the plate boundary fault, and the occasional large earthquake that represents a sudden slip on the fault. The Mariana subduction zone is one of the most seismically active regions in the world, and the earthquakes it produces can be detected by seismograph networks around the globe. The hydrophones in the trench therefore sit at the center of one of the most geologically active environments on Earth, recording the sounds of a planet in constant, if mostly imperceptible, motion. The sedimentary record of the Mariana Trench floor contains a detailed history of the environments of the Western Pacific over the time period during which the trench has been active. The sediments at the bottom of the Challenger Deep consist of a complex mixture of material from multiple sources. The dominant component is a fine-grained carbonate ooze composed of the skeletal remains of planktonic foraminifera and coccolithophores that sank from the surface ocean above. That carbonate ooze records the history of the surface ocean productivity and temperature over the time period represented by the sediment column, and it is one of the primary archives used by paleoceanographers to reconstruct the climate of the Western Pacific during the Cenozoic. Interbedded with the carbonate ooze are layers of volcanic ash from the eruptions of the Mariana Arc volcanoes, each layer providing a precisely dated stratigraphic horizon that allows the sediment record to be correlated with the geological and volcanic history of the region. The deepest sediments in the Mariana Trench predate the formation of the trench itself. As the Pacific Plate descends into the mantle at the Mariana subduction zone, it carries with it the sediments that were deposited on its surface during the millions of years it spent traversing the Pacific Basin. Those sediments include not just the carbonate ooze of the open ocean, but also the biogenic silica of diatom and radiolarian oozes, the volcanic material from seamounts and oceanic islands that the plate passed over, and the fine-grained terrigenous material transported from the continents by wind and ocean currents. That material is being progressively subducted into the mantle, where it will be transformed by the heat and pressure of the mantle into metamorphic rocks and eventually recycled into new igneous rocks at the surface. The Mariana Trench is, in that sense, not just a feature of the present Earth but a machine for processing the geological history of the Pacific into the raw material of the geological future. The paleontological record preserved in the Mariana Trench sediments has been studied by researchers interested in the evolutionary history of the hadal fauna. The amphipods, sea cucumbers, and other organisms that dominate the hadal zone today did not always live there. They colonized the hadal zone from shallower environments as the trench deepened over geological time, evolving the adaptations that allow them to function at extreme pressures. The timing of those colonization events and the evolutionary changes that accompanied them are recorded in the DNA of the living organisms and in the occasional fossil material preserved in the sediment record. Reconstructing that history requires the integration of molecular phylogenetics, paleontology, and the geological history of the subduction zone, and it is producing insights into how life adapts to new extreme environments on evolutionary timescales. The long-term fate of the organic carbon that reaches the Mariana Trench floor is a question with implications for the global carbon cycle. Some of that carbon is respired by the biological community of the hadal zone and returned to the water column as carbon dioxide. Some is buried in the sediment and eventually subducted into the mantle with the descending Pacific Plate, where it is transformed by the high temperatures and pressures of the mantle into graphite, carbonate minerals, or carbon dioxide that is eventually released at arc volcanoes. The amount of carbon that is subducted and the amount that is respired and returned to the ocean are determined by the balance of biological and geological processes in the trench, and that balance has implications for the long-term carbon budget of the ocean and atmosphere. The Mariana Trench is therefore not merely a passive repository for organic carbon, but an active part of the geological carbon cycle that has been operating for hundreds of millions of years. The rate of sedimentation in the Mariana Trench is much higher than in the open ocean basins surrounding it, because the trench concentrates settling particles from a wide area by acting as a topographic focus for the movement of sediment through the water column. Turbidity currents, density-driven flows of sediment-laden water that rush down the slopes of the trench walls, periodically deposit thick layers of sediment on the trench floor, creating a geological record in which periods of slow background sedimentation are punctuated by rapid depositional events. Those turbidites, as the turbidity current deposits are called, carry material from the continental shelves and the slopes of the volcanic arc into the deepest parts of the trench, bringing with them sediment that originated far from the hadal zone and providing an indirect record of the geological and volcanic history of the surrounding region. The human fascination with the Mariana Trench is as deep, in its way, as the trench itself. The desire to reach the lowest point on Earth, the deepest of deep places, is one of the most persistent themes in the history of exploration, and it parallels the desire to reach the highest point, the farthest point, the most extreme point in every dimension. The Challenger Deep is the deep extreme, the bottom of the world, and the handful of people who have reached it describe the experience in terms that go beyond the scientific, a sense of absolute isolation, of being at a place where the human presence is so anomalous and so temporary that the world seems to be made of something older and stranger than the surface world suggests. The question of what lived in the waters above the Mariana Trench before the trench itself existed is one that geologists and paleontologists are beginning to address through the study of the sedimentary and biological records of the Western Pacific. The Mariana Subduction Zone has been active for approximately 50 million years, and the deep water above the trench has progressively deepened as the subduction zone developed and the surrounding ocean crust cooled and subsided. The organisms that inhabit the Hadal Zone today are the descendants of organisms that were living in the relatively shallow water above the proto-trench 50 million years ago, gradually adapting to the deepening conditions as the trench reached its current depth. Reconstructing that evolutionary history requires the integration of geological models of trench development with the molecular phylogenetics of the living Hadal fauna, a research program that is producing increasingly detailed pictures of how the Hadal ecosystem came to be. The acoustic recording made by Victor Vescovo's team during the 5 Deeps expedition captured sounds at the bottom of the Challenger Deep that included ship traffic, military sonar, and what appeared to be a magnitude 4.8 earthquake occurring in real time while the submersible was on the bottom. Those recordings demonstrated that the Challenger Deep is not isolated from the rest of the world acoustically, even though it is isolated physically. The deepest place on Earth is acoustically connected to the ship lanes of the Pacific, to the military exercises of the navies of the Pacific Rim, and to the geological processes that are reshaping the surface of the planet. It is alone and not alone at the same time. The artistic and cultural resonance of the Mariana Trench in the human imagination reflects its status as the ultimate unknown, the place that is most unlike the world we know. The deep sea has always been a repository of the monstrous and the uncanny in human mythology. The sea monsters of ancient maps, the Kraken of Norse legend, the Leviathan of the Hebrew Bible, all inhabit the unfathomable depths that humans could not reach and therefore could not know. The Mariana Trench, as the deepest and most extreme of those depths, concentrates that cultural resonance into a single point on the map of the world. The reality of the trench, a cold, dark, pressurized environment inhabited by pale translucent fish and enormous sea cucumbers and giant single-celled organisms, is arguably stranger than the monsters of mythology, because it is real. The philosophical significance of the Mariana Trench as a place where the conditions of the early Earth may be preserved or echoed is not lost on researchers who study it. The deep ocean, and the Hadal Zone in particular, represents a remnant of the ancient ocean environment, isolated from the surface world by tens of millions of years of evolutionary divergence and by the physical conditions that make it inaccessible to organisms adapted to the surface. The organisms living there are, in a sense, refugees from an older world, survivors of a deep evolutionary lineage that has maintained itself in the darkness while the surface world above went through successive waves of extinction and diversification. The technical challenges of studying the Mariana Trench will not be fully solved by any single technological development. The extreme pressure requires vehicles and instruments built to tolerances that push the limits of current materials science. The remoteness of the location, in the Western Pacific far from major scientific centers, makes logistics difficult and expensive. And the vast extent of the trench, 2,500 kilometers long and containing multiple deep basins with different characteristics, means that any single expedition can sample only a tiny fraction of the total environment. The Mariana Trench will require decades of sustained scientific effort to characterize adequately, and that effort will require the collaboration of researchers from many nations and many disciplines. The most recent discoveries from the Mariana Trench have added entirely new dimensions to the scientific understanding of Hadal environments. The detection of significant quantities of methane in the water column of the Mariana Trench, first reported by researchers studying the fluid chemistry of the trench in the 2010s, opened a new line of investigation into the carbon cycle of the Hadal Zone. Methane, a potent greenhouse gas, can be produced in deep-sea sediments by the metabolic activity of methanogenic archaea, microorganisms that obtain energy by producing methane from carbon dioxide and hydrogen. The high concentrations of organic carbon reaching the trench floor provide ample substrate for methanogenesis, and the methane produced in the sediments may be an important energy source for the chemosynthetic communities of the trench floor. The fate of that methane, whether it is consumed by methane-oxidizing bacteria before it can reach the water column or whether it accumulates and eventually escapes to the ocean above, is a question with implications for the global methane budget and for the understanding of the carbon cycle in extreme environments. The discovery of active mud volcanoes on the floor of the Mariana Trench forearc has added a new geological dimension to the exploration of the region. Mud volcanoes are features where sediment, water, and gas are expelled from the seafloor under the pressure of the fluids released from the subducting slab. The mud volcanoes of the Mariana forearc, some of which rise several kilometers above the surrounding seafloor and extend for tens of kilometers across, are the largest known examples of this geological feature. The fluids they expel are alkaline, enriched in methane and hydrogen, and cold relative to the surrounding water, and they support unusual chemosynthetic communities that are distinct from those at more typical hydrothermal vents. The documentation of deep-sea organisms by the cameras of remotely operated vehicles and landers has produced some of the most striking footage in the history of natural history filmmaking. The images of xenophyophores and holothurians and Hadal snailfish from the Mariana Trench have been seen by millions of people through documentary films and online sharing, creating a level of public familiarity with Hadal organisms that would have been unimaginable before the era of high-definition deep-sea cameras. That visual documentation has had an important effect on the public understanding of the deep ocean as a biologically rich and scientifically important environment, challenging the older view of the deep sea as a barren void and providing the emotional and aesthetic case for its conservation. The strange beauty of the Hadal organisms, their pallor and their translucency and their alien forms, is one of the most effective arguments for taking the biology of the deep ocean seriously. The thermal structure of the water column above the Mariana Trench reveals a complex layering that reflects the different water masses converging in the western Pacific. The surface layer, above approximately 100 meters, is warm and well mixed by wind and surface currents. Below the surface layer, the thermocline, a zone of rapidly decreasing temperature, extends to approximately 1,000 meters. Below the thermocline, the deep water is cold and relatively uniform in temperature, drawn from Antarctic bottom water and North Pacific intermediate water that have spread northward and westward from their formation sites. The deepest water in the trench, below the sill depths that separate the deep basins from the surrounding ocean, is modified by the biological and chemical processes within the trench and has a distinctive temperature and chemical signature that reflects its long residence time within the trench basins. The research into the viral ecology of the Hadal zone is a frontier area that is producing surprising results. Viruses are the most abundant biological entities in the ocean, outnumbering bacteria by approximately an order of magnitude, and they play a crucial role in the ocean carbon cycle by lysing bacterial cells and releasing their contents into the dissolved organic pool. In the Hadal zone, viruses have been found in abundances comparable to those in shallower environments, and their diversity appears to be high. The viruses of the Hadal zone infect not just bacteria but also the amphipods and other multicellular organisms that inhabit the trench, and understanding the ecology of those virus-host interactions is essential for understanding the functioning of the Hadal ecosystem as a whole. The genomic analysis of the microorganisms of the Mariana Trench has produced a library of genes encoding pressure-adapted enzymes, lipid biosynthesis pathways, and other molecular adaptations that are of considerable interest to the biotechnology industry. Enzymes that remain active at high pressure and low temperature have potential applications in industrial processes that are currently constrained by the need for elevated temperatures to achieve adequate reaction rates, and the genes encoding those enzymes could potentially be incorporated into industrial organisms to produce biocatalysts for cold pressure applications. The Mariana Trench is therefore not only a scientific and philosophical frontier, but also a potential source of molecular tools that could have practical applications in the chemical and pharmaceutical industries. The governance of the Mariana Trench as a protected area reflects the challenges of managing resources in international waters beyond the jurisdiction of any single nation. The Mariana Trench Marine National Monument was established by President George W. Bush in January 2009, protecting the waters and submerged lands around the Mariana Islands, including the Mariana Trench itself, from commercial fishing, resource extraction, and other potentially harmful activities within the United States Exclusive Economic Zone. The monument covers an area of approximately 246,000 square kilometers, making it one of the largest marine protected areas in the world, and it includes not only the trench but also the unique biological and geological features of the Mariana Arc and the deep water ecosystems of the region. The protection afforded by the monument, however, applies only within the U.S. Exclusive Economic Zone. The deepest portions of the Mariana Trench, including the Challenger Deep, lie in international waters beyond the jurisdiction of any single nation, and are governed by the United Nations Convention on the Law of the Sea and the International Seabed Authority, which manages the resources of the international seabed. The framework for managing human activities in the international portion of the trench is therefore less complete than for the monument area, reflecting the general difficulty of governing activities in areas beyond national jurisdiction. The scientific community has consistently advocated for the protection of the Hadal Zone from potentially damaging activities, including deep-sea mining, which could disturb the delicate sedimentary environments that preserve the biological and geological records of the Hadal ecosystem. The polymetallic nodules and cobalt-rich crusts that are the targets of most deep-sea mining proposals are found at shallower depths than the Hadal Zone, and the Hadal Zone itself is not currently a target for mineral extraction. But the disturbance of deep-sea sediments by mining activities at shallower depths could produce plumes of suspended sediment that travel to greater depths and affect Hadal communities, and the precautionary management of those potential impacts requires an understanding of the Hadal ecosystem that is still being developed. The monitoring of the Mariana Trench as a baseline for understanding global environmental change is an increasingly important aspect of deep-sea research. The deep ocean, and the Hadal Zone in particular, responds to changes in surface conditions on timescales of decades to centuries, making it a slow but reliable recorder of long-term environmental trends. The temperature, chemistry, and biology of the Challenger Deep today reflect the surface conditions of the western Pacific Ocean over the past several centuries, and future measurements will record the effects of the current period of human-driven environmental change on the deepest and most extreme environment on Earth. The future of Hadal science will be shaped by technologies that are currently in development and that will change the economics and the practicalities of deep-sea research in fundamental ways. Autonomous underwater vehicles with endurance measured in days or weeks rather than hours, capable of conducting systematic surveys of large areas of the trench floor without the constant need for surface ship support, will make it possible to map and characterize the trench on a scale that is currently impossible. Advanced DNA sequencing methods applied to environmental samples will reveal the full diversity of the microbial and myofaunal communities without the need to cultivate organisms in the laboratory. And continuous monitoring stations anchored to the trench floor, transmitting data acoustically to the surface, will provide the long-term records of physical, chemical, and biological conditions that are needed to understand how the Hadal ecosystem changes over time. The Mariana Trench of the next decade will be far better understood than the one we know today. The experience of descending to the bottom of the Mariana Trench, as described by the handful of people who have made that journey, is one of the most extreme sensory and psychological experiences available to a human being. The first thing that changes during the descent is the light. Within the first few hundred meters, the blue of the surface ocean fades to a deep, rich indigo, and then to a blue-black that is barely distinguishable from darkness. By 500 meters, the light is so faint that the human eye can barely detect it. By 1,000 meters, it is gone entirely, replaced by the occasional flash of bioluminescence from passing organisms. The rest of the descent, 10 more kilometers of it, occurs in absolute darkness, punctuated only by the instruments and lights of the submersible. The pressure increase is not directly perceptible to a person inside a correctly designed pressure sphere. The sphere itself is designed to maintain an internal pressure of 1 atmosphere regardless of the external pressure, and the human occupant experiences no physical sensation from the increasing weight of water above. What is perceptible is the compression of the sphere itself. The steel or titanium or acrylic walls flex slightly under the external pressure, producing sounds that can be alarming to an inexperienced occupant. Those sounds, described as pops and cracks and occasional groans, are the voice of the material accommodating itself to pressures that are at the edge of what the engineering allows. The time. The descent takes several hours, and there is very little to see through the porthole except the occasional flash of bioluminescence and the instruments. The occupants of deep-sea submersibles spend most of their time waiting, monitoring the instruments, and thinking. The psychological effects of that prolonged waiting in a small confined space at the bottom of the ocean are reported to be variable. Some people find it profoundly meditative, a forced quietness in which the only relevant reality is the small sphere of light and life within the submersible. Others find it claustrophobic and exhausting. The mental preparation for a deep-sea dive is as important as the physical preparation, and the psychological screening of submersible pilots and scientists for deep-sea missions mirrors in some ways the psychological screening for spaceflight. The geological record of the Mariana subduction zone extends back approximately 50 million years, to the Eocene epoch when the Pacific Plate first began subducting beneath what would become the Mariana Plate. Before that time, the region was occupied by a different plate configuration, and the Western Pacific was the site of a different set of tectonic processes. The initiation of subduction at the Mariana Trench, which may have been triggered by the collision of India with Eurasia and the associated reorganization of Pacific Plate tectonics, produced the beginnings of the volcanic arc that would eventually build the Mariana Islands and created the first deep depression in the seafloor that would evolve into the modern trench. The current extreme depth of the Challenger Deep is therefore the product of 50 million years of progressive deepening as the Pacific Plate continued to subduct and the trench evolved its present character. The moment of landing on the bottom of the Challenger Deep is, by all accounts, anticlimactic in its visual simplicity and overwhelming in its conceptual weight. The sediment, pale and flat, extends to the edges of the lights. There are no dramatic geological features, no towering cliffs, no spectacular landscape. There is simply the bottom, the flat pale sediment of the deepest place on Earth, and the awareness that the column of water above is 11 kilometers deep and that no human technology has ever placed a person in a more remote or more extreme location on this planet. The scientific questions that remain unanswered about the Mariana Trench are more numerous than those that have been answered. The complete mapping of the trench floor, which requires multi-beam sonar surveys at a resolution sufficient to reveal the full complexity of the terrain, is still incomplete. Large sections of the trench walls and floor have been mapped only at low resolution, and the detailed topography of those areas, including the locations of potential hydrothermal vents, mud volcanoes, and other geological features, remains unknown. The cost and logistical difficulty of conducting high-resolution surveys in such remote and deep water has limited the pace of mapping, but new autonomous underwater vehicles with extended endurance and improved sonar systems are beginning to make comprehensive mapping more achievable. The geophysical surveys of the Mariana Trench have revealed details of the internal structure of the subducting slab that are relevant to understanding the mechanics of subduction and the generation of tsunamis. The bending of the Pacific Plate as it begins its descent into the mantle creates a pattern of faults in the outer rise of the trench, the region of the seafloor that is being flexed upward slightly by the bending of the plate before it descends. Those outer rise faults, which are normal faults produced by the tensional stress of the bending plate, can themselves produce earthquakes of considerable magnitude. The outer rise earthquakes associated with the Mariana Trench have been documented by global seismograph networks, and their focal mechanisms provide information about the stress state of the bending Pacific plate that constrains models of the subduction process. Understanding those outer rise earthquakes and the faults that produce them is also important for tsunami hazard assessment, because the vertical displacement of the seafloor associated with outer rise earthquakes can generate tsunamis that propagate across the Pacific. The biodiversity of the Mariana Trench is still being documented. Each expedition that deploys biological traps or sediment corers in the trench returns with specimens that include species new to science, and the true number of species in the Hadal Zone is almost certainly far larger than the current catalogs suggest. The majority of Hadal species are microscopic, and the techniques required to characterize microbial and myofaunal diversity, the diversity of organisms in the size range between bacteria and macrofauna, require sophisticated molecular methods that are only now being applied systematically to Hadal samples. The microbial diversity of the Challenger Deep sediments, in particular, is likely to be orders of magnitude greater than current estimates, based on the general principle that microbial diversity in the ocean far exceeds what can be detected by conventional culturing methods. The connectivity between the Hadal fauna of the Mariana Trench and the Hadal fauna of other ocean trenches is a question that has important implications for understanding how organisms colonize and persist in Hadal environments. The Hadal Zone is discontinuous. The world's trenches are separated from each other by vast distances of shallower ocean, and the organisms adapted to the extreme conditions of the Hadal Zone cannot simply disperse through the shallower water between trenches. Understanding how those populations are connected, whether there is gene flow between the Mariana Trench populations and those of other trenches, and whether the same species are found in multiple trenches or whether each trench has its own endemic fauna, requires the comparison of genetic data from organisms collected in multiple Hadal locations around the world. The long-term changes in the Hadal ecosystem in response to climate change are one of the most important and most uncertain areas of Hadal science. The deep ocean is connected to the surface through the thermohaline circulation, and changes in surface conditions produced by warming, ocean acidification, and changes in primary productivity will eventually propagate to the Hadal Zone on timescales of decades to centuries. The warming of Antarctic bottom water, which is the source of much of the water in the Challenger Deep, is already detectable in the deep ocean, and the implications of that warming for the organisms adapted to near-freezing conditions are being studied. The acidification of the deep ocean, driven by the dissolution of additional carbon dioxide in increasingly acidic seawater, is also a concern for Hadal organisms whose biochemistry is finely tuned to the current pH of the deep ocean. Tonight, as you prepare to sleep, the Mariana Trench is doing what it has done for the last 10 million years. The Pacific Plate is descending into the mantle at a rate of a few centimeters per year, pulling the seafloor of the trench deeper by millimeters over human lifetimes. The amphipods are moving through the darkness, tracking the chemical gradients that lead them to organic matter settling from the surface 11 kilometers above. The Hadal snailfish are hunting, their pale translucent bodies moving through the cold with unexpected agility. The xenophyophores are extending their structures across the sediment, millimeter by patient millimeter. The bacteria in the sediment are metabolizing, cycling carbon and nitrogen and sulfur through the chemical transformations that maintain the chemistry of the Hadal ecosystem. And in the Challenger Deep, at the very bottom of the world, the pressure of 11,000 atmospheres is compressing everything into a density and a silence that is unlike anything else on the surface of the Earth. The human presence in that environment is measured in hours and minutes across the entire history of deep-sea exploration. The total time that human beings have spent at the bottom of the Challenger Deep, across all dives by all submersibles since 1960, is probably less than a day. The trench has been actively subducting for 10 million years. The ratio of human presence to trench history is so small that it cannot be expressed in any units that feel meaningful. We are the most transient of visitors to the deepest of places, and the trench does not notice us. And yet we know it is there. We have measured it, sampled it, photographed it, recorded it, analyzed it. We know that snailfish hunt in its darkness, and that sea cucumbers carpet its floor, and that giant single-celled organisms build complex shelters from sediment particles at its deepest point. We know that its water is contaminated with the industrial chemicals of a civilization that has never seen it. We know that it is connected, acoustically, to every ocean and every nation. We know that it is old and cold and dark and extraordinarily alive. The strangeness of the Mariana Trench is not reducible to the extremity of its physical conditions. Its strangeness is that it exists at all, that the processes of plate tectonics, which are themselves the product of the internal heat of a cooling planet, have produced a place this deep and this dark and this remote, and that life, which appeared on Earth approximately 4 billion years ago and which has never found a place too extreme to colonize, has made itself at home there. The universe does not tend toward comfort. It tends toward complexity, and complexity tends toward life, and life tends toward the available energy in whatever form it takes, whether that energy comes from the sun or from the chemistry of deep rocks or from the organic material drifting down through 11 kilometers of water. The isolation of the Mariana Trench from other hadal environments around the world has produced distinctive evolutionary trajectories in the organisms that inhabit it. Comparing the fauna of the Mariana Trench with the fauna of other Pacific trenches, such as the Kermadec Trench to the south, the Tonga Trench, and the Japan Trench to the northwest, reveals both similarities and differences that reflect the combined effects of dispersal between trenches and isolation within them. Some amphipod species are found in multiple Pacific trenches, suggesting that they are capable of dispersing across the barriers of shallow water between trenches, perhaps through the intermediate depths of the deep sea basins. Others are apparently endemic to a single trench, having diverged from their relatives through the combined effects of genetic isolation and adaptation to the specific conditions of their particular hadal environment. Disentangling those dispersal and isolation histories requires the comparison of molecular data from organisms collected in multiple trenches, a research program that is gradually producing a picture of how the hadal biosphere is connected and partitioned. Sleep now, in the warmth and the light of your ordinary world, knowing that 11 kilometers below the surface of the Pacific Ocean, in a darkness that has never been touched by sunlight, life is continuing its ancient business. Not because it is trying to. Not because it has a purpose. But because that is what life does. It finds a way. It always finds a way. The ocean is patient. The trench is patient. The life at the bottom is more patient than either. It has been there for millions of years, adapting to pressures and temperatures and chemical conditions that change only on geological timescales. A human lifetime, in that context, is less than a blink. And in that blink, we have managed to reach the bottom and look, and what we have seen has been worth every moment of the extraordinary effort required to see it. The social behavior of the hadal amphipods is one of the most unexpected findings of deep-sea biology. Animals living at such extreme depths, in an environment where food is scarce and widely dispersed, might be expected to be solitary and to minimize energetic expenditure. But the lander camera footage from the Mariana Trench consistently shows amphipods aggregating in dense swarms around bait, with hundreds or thousands of individuals converging on a food source within minutes of its detection. That aggregation behavior, which requires the ability to detect chemical gradients in the water and to move rapidly in a coordinated fashion, implies a level of sensory capability and behavioral sophistication that was not anticipated from organisms living in such extreme conditions. The mechanisms by which amphipods detect food at distances of potentially hundreds of meters in the hadal environment, and the chemical signals they use to communicate the presence of food to conspecifics, are areas of active research. The role of the Mariana Trench in the global water cycle is another aspect of its significance that is often overlooked. The subduction of the Pacific Plate carries with it not only the sediments and organic carbon that accumulated on its surface, but also the water that was incorporated into the oceanic crust through hydrothermal circulation and that saturates the sediments overlying the plate. That water, estimated to amount to several 100 million cubic kilometers over the history of the subduction zone, is driven into the mantle by the heat and pressure of subduction, where it plays a crucial role in the generation of arc magmas and in the cycling of volatile compounds between the Earth's surface and its interior. The Mariana Trench is therefore not just a feature of the ocean surface, but a portal through which water, carbon, and other volatile compounds are transferred from the surface environment to the deep interior of the Earth, and through which new volcanic material is returned to the surface in the form of the Mariana Arc volcanoes. The history of the naming of features within the Mariana Trench reflects the history of its exploration by different national scientific programs. The Challenger Deep is named after HMS Challenger, the British naval vessel that first recorded a depth in the region in 1875. The Sirena Deep, a secondary basin in the Mariana Trench that reaches depths of approximately 10,809 meters, is named after the Spanish research vessel that measured it in 1997. Other named features within the trench reflect the vessels or expeditions that discovered them. That naming convention, which is common in marine geography, preserves a record of the exploration history of the trench in the names of its features, encoding the national and institutional affiliations of the scientists who first charted each portion of the deepest ocean. The philosophical and scientific implications of the Mariana Trench for the concept of the biosphere, the total envelope of life on Earth, are profound. The discovery that life exists at every depth in the ocean, from the surface to the deepest trench, and at every depth in the ocean sediments, from the seafloor surface to many meters below it, has radically expanded the estimated volume of the biosphere compared with earlier definitions that emphasized the thin layer of life visible at the Earth's surface. The total biomass of the deep biosphere, including the organisms living in the ocean water column, the ocean sediments, and the oceanic and continental crust, is now estimated to be comparable in magnitude to the biomass of all surface life. Life is not a thin veneer on the surface of the Earth. It is a deep, pervasive presence that extends throughout the accessible portions of the Earth's crust and into the depths of the ocean basins, with the Mariana Trench representing the extreme lower limit of that inhabited volume. The comparison between the biological communities of the Mariana Trench and those of other extreme environments on Earth provides important context for understanding the adaptations required for hadal life. The organisms of hydrothermal vents, cold seeps, hypersaline brine pools, and deep subsurface environments have all evolved biochemical and physiological adaptations to their respective extreme conditions, and those adaptations share some features with the adaptations of hadal organisms while differing in others. The cold and high-pressure adaptations of hadal organisms are distinct from the high-temperature adaptations of vent organisms, but both reflect the same fundamental principle: that life can evolve to function in conditions that are far outside the range tolerated by organisms adapted to the more moderate conditions of the surface world. That principle, which is one of the most important findings of extreme environment biology, has profound implications for the search for life in other extreme environments, both on Earth and beyond. The silica cycle in the Mariana Trench ecosystem connects the biological activity of the trench to the broader biogeochemical cycles of the Pacific Ocean. Diatoms and radiolarians, the primary siliceous plankton of the surface ocean, produce skeletal structures of biogenic silica that sink after their death and dissolve in the undersaturated deep water of the Pacific. In the Mariana Trench, the dissolution of those siliceous skeletons on the trench floor and in the sediments is accelerated by the high pressure and low temperature, which increase the solubility of silica and the rate of dissolution reactions. The dissolved silica released by that dissolution process enriches the trench water relative to the surrounding ocean, creating a chemical signature that oceanographers can use to trace the circulation of water within and around the trench. That enrichment in dissolved silica also affects the biological community of the trench, because silica is an essential nutrient for the production of diatom and radiolarian skeletons in the overlying water column. The light produced by bioluminescent organisms in the water column above the Mariana Trench is one of the most extraordinary biological phenomena in the ocean, though it is invisible to the casual observer at the surface. The deep scattering layer, a dense aggregation of bioluminescent organisms that migrates vertically between depths of several hundred meters and several thousand meters on a daily cycle, produces enough light in the deep twilight zone to be detectable by extremely sensitive photomultiplier tubes lowered into the water. The organisms producing that light include lanternfish, firefly squid, dinoflagellates, and many other species, each using their bioluminescence for different purposes. To lure prey, to avoid predators, to communicate with conspecifics, or as a byproduct of chemical reactions with no known function. That biological light is the only illumination in the deep ocean below the photic zone, and it shapes the sensory world of the deep-sea organisms that inhabit those depths. The monitoring of the Mariana Trench for evidence of past and present tectonic activity has produced important information about the dynamics of the Pacific Plate and the potential for large earthquakes in the region. The GPS measurements of the Mariana Islands, which sit on the overlying plate, show the slow deformation of the overriding plate as it responds to the drag exerted by the subducting slab and to the periodic stress releases associated with large earthquakes on the plate boundary fault. That deformation, which accumulates over decades between large earthquakes and is released in sudden jumps during earthquakes, provides information about the state of stress on the plate boundary and about the potential for future large earthquakes. The Mariana subduction zone has historically produced earthquakes of magnitude 7 and higher, and the monitoring of the tectonic deformation of the region is an important input to the assessment of seismic and tsunami hazard for the islands and coastlines of the Western Pacific. The daily vertical migration of the organisms in the water column above the Mariana Trench creates a biological conveyor belt that connects the sunlit surface to the dark depths. Organisms that feed in the rich surface waters at night descend to dark depths during the day to avoid visual predators, carrying the organic carbon they have ingested in their bodies. When those organisms die, their bodies sink and eventually reach the trench floor, contributing to the organic input that sustains the Hadal community. That biological pump, which transports carbon from the surface to the deep on timescales of days to weeks, is far faster than the physical sinking of individual particles, and it represents a significant pathway for carbon export from the surface to the deep ocean. Understanding the magnitude of that biological pump in the water column above the Mariana Trench and how it varies seasonally and interannually is important for understanding the carbon cycle of the Western Pacific and for modeling the ocean's role in the global carbon budget. The study of the Mariana Trench has also contributed to the development of new materials and engineering solutions that have applications far beyond ocean science. The pressure vessels used in deep-sea submersibles, which must maintain structural integrity at pressures that would crush conventional steel structures, have driven innovations in high-strength alloys, in the design of transparent materials such as the acrylic viewports of modern submersibles, and in the development of buoyancy materials such as syntactic foam that can maintain their structural properties at extreme pressures. The pressure-resistant seals, valves, and connectors used in deep-sea instruments have informed the design of similar components used in aerospace, in subsea oil and gas production, and in other industries where extreme pressure resistance is required. The engineering challenges of the Hadal Zone have therefore had a productive fertilizing effect on materials and engineering science that extends well beyond the immediate application of reaching the deepest ocean. The experience of watching deep-sea footage from the Mariana Trench, even on a screen, produces a distinctive psychological effect that is different from watching wildlife footage from surface environments. The darkness, the cold blue-white light of the camera systems, the slow movements of the organisms, and the awareness of the crushing weight of water above create a sense of an environment so far removed from everyday experience that it approaches the alien. That alienness is not uncomfortable but fascinating, a reminder that the world contains environments of extraordinary strangeness that exist independently of human awareness and that will continue to exist long after human civilization has passed from the scene. The Mariana Trench does not need to be known to exist. It has existed for 50 million years without our knowledge, and it will exist for 50 million years more whether or not we continue to explore it. Our knowledge of it changes us, not it. The recovery of intact pressure vessels from deep-sea missions gone wrong has provided unexpected scientific data alongside the engineering lessons. Several deep-sea landers have been lost during deployments in the Mariana Trench, either because the acoustic release mechanism failed to respond or because the ascent line was severed by the trench walls. Some of those lost landers have subsequently been recovered by other expeditions, having spent months or years on the trench floor before being found and retrieved. The biological and chemical data from those extended deployments, representing exposure times far longer than any planned experiment, have provided unique information about the long-term changes in the bait and the biological community around it, and about the rates of corrosion and material degradation at extreme depth over extended periods. Those accidental long-duration experiments have contributed to the understanding of the hadal environment in ways that would have been difficult or impossible to design deliberately. The comparison between the Mariana Trench and the other great trenches of the Pacific Ocean illuminates the ways in which the local geology and oceanography shape the character of hadal environments. The Tonga Trench, the second deepest in the world at approximately 10,800 meters, differs from the Mariana Trench in its higher rate of subduction, its younger subducting plate, and its position in a region of very high biological productivity in the surface waters above. The Kermatic Trench, to the south, sits beneath the highly productive waters of the Chatham Rise and the Antarctic Circumpolar Current, and its hadal fauna is shaped by the higher input of organic material from above. The Japan Trench, which was devastated by the 2011 Tohoku earthquake and tsunami, provides a dramatic example of how major geological events can rapidly transform the biological community of a hadal environment. Each trench is a unique experiment in hadal ecology, shaped by the particular combination of geological, oceanographic, and biological factors that characterize its location. The Mariana Trench is, in the end, a place that changes the way you think about the word impossible. Impossible pressure. Impossible cold. Impossible darkness. Impossible depth. And yet life, which has been developing strategies for surviving impossible conditions for 4 billion years, is there. It found the energy sources. It evolved the molecular tools. It adapted the membranes and the enzymes and the cellular architecture. And it continues, patient and dark and cold and utterly indifferent to the impossibility of its own existence. That is what life does. It does not ask permission from the laws of physics. It finds the exceptions. It exploits the gradients. And it persists, long past the point where persistence seems reasonable, because persistence is the only strategy that life has ever truly needed. The data recovered from the deepest parts of the Mariana Trench has also forced a reconsideration of the models used to estimate global rates of carbon burial and remineralization. The conventional models assumed that most of the organic carbon reaching the deep ocean floor was rapidly remineralized by bacterial decomposition, with only a small fraction being buried in the sediment for geological timescales. The high rates of organic carbon input to the Mariana Trench, concentrated by the funnel-shaped topography, and the biological community that processes that input, suggest that hadal environments may be sites of disproportionately high carbon burial relative to their area. If hadal environments account for a larger fraction of deep-sea carbon burial than previously assumed, the global estimates of carbon sequestration in deep-sea sediments may need to be revised upward, with implications for the models of the global carbon cycle that are used to project future atmospheric carbon dioxide concentrations. The sounds that humans have introduced into the Mariana Trench, from the acoustic transponders used to communicate with submersibles and landers to the sonar systems of the survey ships overhead, are themselves a form of environmental perturbation that has received little attention. The deep ocean is full of natural sound, from the geological to the biological, But the frequencies and intensities of anthropogenic sound are different from natural sources in ways that may affect the behavior of deep-sea organisms. The amphipods that aggregate around baitlanders, for example, may use acoustic cues to detect the presence of food from distances that exceed the range of chemosensory detection, and the introduction of novel sound sources into the hadal environment could potentially interfere with those behaviors. As the exploration of the hadal zone increases in intensity, the acoustic footprint of that exploration will grow, and the potential effects on the organisms of the hadal environment will need to be considered alongside the potential effects of other forms of disturbance. If this documentary kept you company tonight, subscribe to the channel. A new world every week, always deeper, always stranger. The bottom of the ocean is waiting. And so is the next question that no one has thought to ask yet.